Good morning, everyone. Good afternoon and good evening to everyone joining us today, and welcome to today's webinar. Today's talk is entitled, Does Real-World Evidence Play a Role in Precision Oncology? My name is Kelsey Brown, and I am the Director of Metal Coating Solutions at TransPerfect Life Sciences in London, and it's my pleasure to be your moderator for today's session. Today's event will run for approximately 60 minutes, including time for Q&A with our speakers at the very end. It's interactive and it works best when you're involved, so please feel free to submit questions and comments for our speakers by using the questions chat box in your control panel, and then we'll answer your questions during the Q&A session. At this time, all participants are in listen-only mode, and if you do require assistance, please feel free to contact me at any time by sending me a message using your control panel. Please note the session will be recorded and made available to you on the IDDI website for viewing within 24 hours after the webinar is complete. This event is proudly hosted by the International Drug Development Institute, IDDI. IDDI is an expert center in biostatistical and integrated eClinical data services for pharmaceutical and biotechnology companies in several disease areas, including oncology and ophthalmology. IDDI optimizes the clinical development of drugs, biologics, and devices thanks to proven statistical expertise and operational excellence. And founded in 1991, IDDI is headquartered near Brussels, Belgium, with the U.S. Center of Operations in Raleigh, North Carolina. Now I will pass this over to Dr. Mark Busey, the Chief Scientific Officer and founder of IDDI, who will introduce today's speaker.
Thank you, Kelsey. It gives me great pleasure to introduce the speaker today, Dr. Hobbs, who has a degree in biostatistics from the University of Minnesota and has worked much of his career in Texas. He's currently at the University of Texas. He also worked in the Cleveland Clinic, and he has received a number of prestigious awards. You can take a look at his CV with his abstract for this webinar. He has, I think it's fair to say, he has a lot of experience and he is recognized as a leading expert in the design of clinical trials. So it's really a pleasure to have him today to discuss a subject that I find particularly enticing and also somewhat frightening for reasons that will become clear. And I think he's probably the best person to talk about the role, if any, of randomized evidence in registration of new drugs, and so the complementary role of real-world evidence and clinical trials in registrations in new drug applications to the agencies. So Brian, I'm really delighted that you accepted to give this webinar, and we all look forward to your presentation. There will be 5 to 10 minute questions at the end of your presentation. The floor is yours. Thank you again.
Okay, excellent. So please confirm that you can see my screen.
We can. Yes, we can see it very well now.
Okay, excellent. I'm going to minimize these control panels. Can you see those? Or do I just see those?
That's just visible to you.
Oh, okay. I think hopefully there's a way to minimize that. Oh, yes. Okay, excellent. Okay, so I'd like to thank Dr. Boyce for the opportunity to give this presentation. And thank you all for joining today. I'm always flattered that people want to hear me speak. So thank you very much for your time. As Dr. Boyce said, I started my career at MD Anderson Cancer Center, where I was really right in the heart of adapting methodology for precision oncology, both in design and analysis. And then I had the opportunity to start the cancer biostat group at Cleveland Clinic. So a lot of this talk is going to be, people have asked Brian to do things in this space, and this talk is a culmination of things I've tried to do, also things I've learned that perhaps we cannot do. So I want you to think about it from that perspective. I've also co-founded a company called Telperion that builds technology, and I can talk a little bit about that if you're interested as well. I have some disclosures. I have been an advisor for several companies. I'm currently advising one of the FDA grants that was awarded to Genentech on this topic, actually exploring the use of real-world data to generate real-world evidence and regulatory decision-making. Like I said, I co-founded a company in 2018 that's really bringing computer scientists and software developers into statistical methodology. I haven't seen that combination yet, and we've had, we build tools basically for clients. So I'll get started here. If you have looked, one caveat, if you have looked for me on social media, you probably haven't found me because I, as a scientist and as somebody that's involved in a lot of these discussions about evidence, I've just never felt like I wanted to engage in the discussions that I see happening about drug development on social media. So actually, if you do see me on social media, maybe you should let me know, because it's probably not me. I do have a LinkedIn page. It needs to be updated. Probably 8 years ago, I created it. So I'm not very active on social media at all. If you do want to find me, the best way is to email me at my University of Texas address. Okay, so before we start, we're gonna poll the audience here. So I really enjoy this show that's in the US on NPR called Intelligence Squared. And they do this thing called Oxford style debating. They ask the audience a question, and it's usually a yes or no question. And then they debate, and then they compare the results at the end, and they see kind of which side has moved the needle the most relative to baseline. So we wanted to do that here, and I thought Dr. Boyce kind of encouraged that we do something like this, so I think it's a great idea. So the motion to you is, owning to the challenges that confront us as we develop these targeted therapies, adoptive cellular therapies, immunotherapies, given the challenges with these rare subpopulations, will real-world evidence play a transformational role in the future of registrational trials for precision oncology settings? So you get to respond as you agree with this statement, you disagree with this statement, or you do not know, you abstain from a decision. So I think, should I pause here while everybody votes, or do I go ahead with the presentation? What do you guys think?
I think you can give people, let's say, 15 seconds to think about the question. Okay. And I was trying to vote, but I just see that organizers and panelists may not vote, so I'm getting frustrated. But anyway, everybody else can vote.
Maybe hold your vote, Mark, and you can. Or what do you want to do? Will you be swayed by the evidence here? Will you possibly change your vote?
Absolutely. Why not? Absolutely. Open-minded. Let's move to the results if we could. Well, I think people have had time to vote, hopefully. Oh, wow. Do you see this, Brian? I don't think you see the results, actually. So let me read the results to you. 45% of the participants agree that real-world evidence will play a transformational role. 23% disagree, and 32% don't know. So I hope at least that the last percentage don't know will perhaps move after your presentation.
Okay, excellent. Thank you for participating. I think this is great. And that's kind of consistent with, I think, my expectations. So as I went back, okay, so let's get into it. As I was going back and, I realized that when I'm asked to speak about this topic, it's usually, I have stopped talking about the detailed methodology and I've started to talk about the context. This is going to be a contextual talk, a conceptual talk. I'm not going to get into details of specific models. I love doing that. So if there's interest in that, I can talk with you offline, but that's not going to be the discussion today. And additionally, I realized as I was going back through the slides that this is heavily influenced by regulatory policy within the United States. So I'm focused a lot on FDA policy in this presentation. So I apologize for not integrating some of the EMA and MHRA policy, and that's something I need to do in the future. So, okay, so real world evidence. If you've been around at all the last decade, you've seen the kind of monetization of this idea of real world evidence. And I think this diagram from CancerLink is an excellent illustration. We started to see this kind of thing, I would say 2016-ish. I have worked with CancerLink at Cleveland Clinic. I know Elizabeth Garrett Mayers personally, and she's an excellent researcher. But if you see the marketing departments of all of these places kind of creates these diagrams that look like this, kind of selling, this idea that real world evidence is a very broad thing. It encapsulates consumer data, randomized trials, pharmacy data, also shows social media is in here, right? And what we often think about in a research context is electronic medical health records. Medical health records are, collected for the purpose of treatments and billing, not necessarily for the purpose of treatment. For the purpose of research. So we see these diagrams, we see real world evidence, and we see people placing all of this into one kind of larger schema, right? And perhaps social media could be useful, but I think actually if we're going to use these, we would have to think about specific methodological context and frameworks for using them. They're not all one thing. Maybe social media is helpful for sentiment, right? If you're a large pharma and you need to evaluate sentiment about a drug. It's not clear that all of these or any of these will be useful in a registrational trial. So the reason I get asked to talk about this a lot is because I did develop methodology a long time ago that considered how we integrated kind of historical control arms into trials and potentially adapt those trials in the presence of bias. Now, I was doing this in the context of cooperative trials in the United States. It was being done out of Mayo Clinic. The Mayo Clinic was running one of the cooperative groups, and they actually did have trials where they did repeat the control arms. The control arms in CRC, for example, didn't change a lot over a sequence of studies. We had the FOLFOX and the FOLFIRI regimens, and there was an honest, Dan Sargent, who was one of my thesis mentors, had the interest in how do I leverage this information? It's the same sites, it's the same regimens. So we did, part of my thesis was thinking about this. One thing to note here, if we have this Kaplan-Meier curve at the top, which represents a historical trial, and then we have a concurrent randomized trial here, where we randomize some patients to control, basically potentially replicating this information, I want you to look at the right side. The reason this is controversial is that you can't just pool the data. In fact, the bias is, it destroys the treatment effect, the inference of the treatment effect. So I'm showing you on the x-axis here, the magnitude of treatment effects, lambda here. I'm calling at 0, there's no treatment effect. So if we follow this vertical line at 0, there's no treatment effect. Now this power surface should be vertical. As we have more treatment effect, we have more power, but you can see it's highly disrupted by the bias. This bias is the difference between the historical control and the current control. If the current control is better, we detect treatment effects that don't actually exist with 80% power here. Even harmful treatments look good. On the other hand, if the historical control, outperforms the concurrent control, we lose our power. We have effective treatment, we don't have power to identify it, right? So this is a fundamental issue with why this is challenging. We can't just pool this data. So I did think about frameworks for integrating this in hierarchical modeling, evaluating extent of bias, mapping the extent of information gained into an effective sample size, and then thinking about kind of optimal reallocation of the rest of the trial. Idea being, if there's substantial bias, you would continue with a 1-to-1 randomization ratio. If there was limited bias, you could tweak that randomization ratio, and you could kind of use more of the remaining patients for the experimental arm, trying to get balance at the end of the trial. So I had this idea a long time ago, and since then I've been asked to talk about this quite a bit. And this question, does this apply to real world data? Remember, this was being done in the context of cooperative trials run at the same sites. Those are clinical trials, those historical cohorts. You can see if you do this adaptive design, we can correct this power surface. So methodology can help us in this setting. We don't have this diagonal line anymore. We do have disruption of the power surface, but with substantial bias, it goes back to as if you didn't use the data. So I think there are things you can do from a methodological perspective. If we fast forward now, these ideas are actually being used in trials in the United States. So Genentech has used this in one of the complex innovative trial designs, one of the first 5 that the FDA initiated in lymphoma. And they're doing it for patients that actually have protocol violations. patients that are randomized and then drop off, they continue to follow and they use them as an external control cohort. And that borrowing actually facilitates their inference of survival in a setting where they wouldn't really have enough information to evaluate survival. So, you know, this is being used, and I think, so I'm aware of what's going on too in this development because I'm advising this grant. So the FDA, okay, so now there's this question, does this apply outside of those contexts that I discussed? So as we take that historical evidence and historical data and we move it into the real world setting, can we apply these methods? And that's what I often get asked. Now the FDA has been heavily, engaged in this topic. There were 31 applications a few years ago for this, exploring the use of real world data to generate regulatory evidence and decision making. They awarded 4 grants. I'm helping advise this, I'm one of the participants in this Genentech UNC grant. So the FDA is funding this. They're trying to get, feedback on this. They have developed kind of 3 buckets where they believe real world evidence has a role from a regulatory perspective. What often I get asked to talk about is this idea of a hybrid control arm, that, we do have trials with rare subpopulations. They are hard to study with traditional randomized trials. Can we do a 4-to-1, 5-to-1 randomization design and supplement that control arm with some external evidence, right? So this is the idea that I think has taken hold the most when we think about the topic today. And we can see diagrams from the FDA that kind of paint a spectrum of how much we rely on real world data. So they talk about on the far left side here, we have traditional randomized trials, and real world data is only used to make statistical assumptions in planning. And then there's this second shift to the right here where we have some pragmatic elements to the trial, and we might integrate external controls, right? And then we get it fully externally controlled, no concurrent controls, and then we have fully observational studies, right? So they're talking about this and setting up these diagrams. Obviously, the hybrid controlled trial is here in this second one. Okay, so a lot of activity has happened in recent years in this space in terms of guidance documents. I think the most important one came out in February, this 2023, considerations for design and conduct of externally controlled trials for drug and biologic products. If you're interested in this topic, you really should read that. Despite all the, discussions from regulators about this topic, there's a caveat here. And that caveat is the basis for approval remains the same. Substantial evidence that all drugs will have the effect, and we need adequate, well-controlled clinical investigations. So I highlighted that because I think that's maybe the, I think this is the crux of the matter. How far away can we get from a well-controlled clinical investigation? How do we define that in the context of real-world evidence? How do we even define it in the context of what we've seen recently with open-label randomized studies in oncology? So I think it all centers around this question. And we see regulators saying that we do need to think about rarity of the disease and lack of suitable control. That's been consistent throughout these discussions. Okay, so we have the FDA interested in this topic, funding research in this topic, but still taking this hard line of we haven't changed the basis of approval. We have the other end of the spectrum where the monetization of real world evidence has kind of just gone, what do you want to say? It has increased and it's increasing over time. And I want to put up this article because I think this is very typical of what you see from this other end of the perspective. Dr. Budi is a computer scientist and physician from UCSF, and Dr. Budi has given TED Talks where he has claimed that we no longer need clinical trials, right? All of the information we need are in these servers, which are stacked behind him, right? He's taking a photo in front of his server stacks, right? This is no longer a biological or clinical question. This is a question of algorithms, right? And the computer scientists in Silicon Valley will find the cures based on data that has already been collected, right? this is the narrative that we see coming out of the kind of Silicon Valley computer science groups. And I can tell you the American public loves this narrative, right? And you can see the hyperboles in these articles. You know, he's looking for biomedical moments and he explores huge data sets, right? The idea being the data sets are huge and the information's there. We just haven't appropriately extracted it. And, you know, I could go on. There's more articles where he makes claims that I don't think are true based on my experience with actually working in precision oncology. You know, and then this pivot from that community to say, this is not a clinical scientific problem. It's a crowdsourcing problem. The solution to this is retroactive crowdsourcing. And, you know, a high school kid can do that. So, you know, we have that narrative coming out. And I think that narrative is looking to monetize these databases, right? Let's be honest. So I came up with my own checklist for what to think about in the context of registrational trials when we think about real world evidence. I came up with this because I was a discussant on this topic at JSM last year, this last few months ago. So this is my checklist on things that I think would be crucial to think about before we can make this consideration of incorporating real world evidence in this setting. The first is acuteness for efficacy. What is the complexity and rarity of the disease, and how infeasible is it to randomize? Right, we have to have those two conditions to go down this path. The second is endpoints. What is the nature of monitoring the disease in the real world setting? And is that reproducible in itself, but is it rigorous and consistent with the way we monitor endpoints in a clinical trial? Prognosis. Has our real world data actually captured things that are essential to understand prognosis? And now I have, you know, elements that are absolutely fundamental in oncology research. We need to know the line of therapy of the patient. Now, if the patient gets treated at multiple centers or they have flown to MD Anderson Cancer Center for a clinical trial, but they were treated initially at their local community clinic, do we know that this is a second line patient or a third line patient? Like, that's absolutely critical. Do we know, you know, when the diagnosis happened? Do we know that the extent of the diagnosis from the community clinic is actually accurate when assessed maybe at an academic medical center? What is the completeness and what is the reliability of the other basic clinical prognostic attributes that we would need to know about the patient? If we're talking about EHR, those are databases that are constructed for the purpose of treatment and billing. Did we adequately define the patient's stage? You know, did we adequately measure their tumor burden at baseline? Were they measured or scanned at all? And then we move into what is precision medicine about, right? It's about novel targets, molecular targets, genetic targets. It's about, you know, extracting T cells from patients, improving them, boosting them, and then reinserting them with the adopted cellular therapies that we see. It's about immune phenotypic characterization of what's going on with the immune response. You know, any of these biomarkers in these databases. That's the third thing. And then at that point, if you can satisfy these first three, then I care about methods. Then I want to know how you're actually going to adjust for bias. Are you going to match? Are you going to discount? You know, what are you going to do about bias and mostly measured confounding? Then I care about that. So I wanted to go through kind of a tale of two diseases here that I'm working with. I've always worked in the oncology context, but I'm also working with the Cystic Fibrosis Foundation now. And I've learned a lot about how different these contexts are. And I think they tell us something, they elucidate this. So cystic fibrosis, a life-shortening multi-organ genetic disease characterized by this lack of oxygen absorption that patients experience. The primary cause of early death is lung function loss. Now, there's been dramatic improvements in treatment of cystic fibrosis. There is a new class of therapies that effectively works for 90% of the CF population. What the industry is looking to do now is address that roughly 10% that are not eligible for these modulators, right? So we are CF is already a rare disease. Now we're talking about a subset of a rare disease. That 10% might be 3,500 individuals in the US every year. So, so for me, there is acuteness for efficacy, very rare disease. There is a need here to integrate external evidence. Let's talk about how patients let's talk about the other elements. Patients are diagnosed at birth in the United States, and there's a registry that follows them. There's over 35,000 patients living with CF that are followed in the registry. This diagnosis is clear, and prognosis is pretty clear because these patients are followed in this registry, and there is a natural decline that is involved with this disease that can be measured. Furthermore, disease progression is monitored universally. So whether you enroll in a clinical trial or you're going into your routine clinical care visit, they're going to do a spirometry measure. And they're going to use this percent predicted FEV1 as the measure for monitoring your disease, completely consistent with the clinical trial. And also measured at regular visits. I was told every 6 months patients get this measure. So the endpoints are satisfied. So this is the case. This is interesting. Now let's pivot to precision oncology, right? I think you could see I've set up a contrast here. Precision oncology is also becoming a rare disease. Cancer in itself is becoming a rare disease. What we've seen in the last decades is a reclassification of cancers from organ system to genetic subtype, immune phenotypic subtype. This is a dated article, maybe 2018, and we see there are somewhere around 25 subtypes now of non-small cell lung cancer. Some of them are quite rare. Now, what we're being told by drug developers as they target specific mechanisms is that these should not be considered a single disease. Like, these are sub-diseases of non-small cell lung cancer with different mechanisms by which the tumor cells are surviving and proliferating, right? So there's definitely acuteness for efficacy because it would be hard to study, a BRCA patient, which is 1.3% of the population. Furthermore, we now have immune phenotypic subclassification, right? A cancer immunity cycle has driven a lot of this, where we understand we can interrogate what's going on with the patient's innate immune response. We can assess something called the immunity set point, which looks at the balance of T cells versus PD-L1 and how much those T cells are being inhibited. And patients have different immune systems that are responding differently to their cancers. So again, you could subset patients by all of these biomarkers that we see that describe the process. So we have acuteness for efficacy. We do see regulatory policy has evolved now because of this. We could when we were looking at single histology, single biomarker studies, we could get conditional approvals after phase 2. We now have tumor agnostic drugs where we run trials that enroll multiple indications, but they have a common genetic or immune phenotypic target. And we have seen accelerated approvals after phase 1 for those studies. I was able to review this landscape for Nature Review's Clinical Oncology with my colleagues at MD Anderson, and we have described basically the development pathways for the 7 tumor agnostic approvals that we have another one now, right? But the first 7 tumor agnostic approvals, where now we're defining these drugs can be administered on the basis of a molecular feature, a genetic feature, or an immune phenotypic feature, right? Not on the basis of an organ class. So this is what's happening in precision oncology, and I want to give you a very brief case study that I think is elucidating for this question, because we would think that, okay, real-world evidence is needed here because we have smaller and smaller subpopulations. It's going to be harder and harder to randomize. Let's look at the Atezolizumab's development in second-line bladder cancer. So they ran a single-arm phase 2 trial after phase 1. This was a label expansion study, so the Atezolizumab had already been approved in other indications. All of these companies have, okay, so this is immunotherapy, and all of these companies had different ways they defined PD-1 high. So Roche Genentech used immune cell staining to define PD-1 high, and they had, in essence, 3 levels of patients. Now, the drug is targeting PD-1 high patients. That's indicated by IC23. It means 10% or more of the immune cells have PD-L1 positivity expression. We also have IC1 and IC0. So those are lower levels of PD-1 expression on immune cells. They ran this trial as an all-comer trial, so they did not restrict the targeted biomarker. And they compared objective response rates across these biomarker subsets. So the target is IC23 or PD-1 high. That population got a 26% response rate. That's about 100. But there's about 200 that are not PD-1 high based on their classification, and we see 11 and 8% response rate. So they told us the historical null was 10%, right? So we're compared to 10%, we got 26. They give us Kaplan-Meier curves that show the biomarker strata. These are not different arms. This is a single arm study, but they're showing that the IC23 population is above, surviving longer than IC1, IC0. Okay, on the basis of that, increased levels of PD-1 expression on immune cells were associated with increased response. So they got accelerated approval in 2016. Let's fast forward to the phase 3 trial that followed. The phase 3 trial is very interesting to me. It randomized 931 patients, but only 234 actually the primary analysis. The primary analysis is going to compare chemotherapy to atezo in this IC23 population, high PD-1. So they enrolled a lot of patients that were not involved in that primary analysis. They did not restrict the trial to the targeted biomarker population. And they found that overall survival did not benefit. The p-value was 0.41. Moreover, objective response rates were similar between IC23 and control. So if we look at this, they got, if we look at the Atezolizumab population in the target, IC23, 116 patients here, they got 23% response rate in this phase 3 study, so pretty close to the 26, right? Recall that the null hypothesis was 10%. Let's look at chemotherapy, however. There's 118 patients, and it's a 22% response rate, not 10%. When we look at all comers and you compare atezo to chemo, we do see they're pretty much identical, right on top of each other. Both had 62 responses, about 13.4% of the population. So, the off-target patients are very close to chemo, they're the same. The on-target patients are also close to chemo, but they're doing better. Chemotherapy is doing better for this biomarker target. That seems to me that was unexpected, right? Or you look at the Kaplan-Meier curves, there's violation of proportional hazards. We do see a heavier tail here for atezo. So there are some patients benefiting, I believe, but it was not significant. Median survival was 11.1 versus 10.6. Now, I gave this story because this is not the only story with precision oncology. This story has been repeated again and again. It also happened with Durvalumab in the same indication. On the basis of this phase 3 trial in 2021, these drugs were removed from accelerated approval. They were withdrawn. We did some digitization of this trial data, and we wanted to answer this question, what's going on? We took both the Danube study, which is Durvalumab, and we took the IMvigor study, which is Atezolizumab. And we wanted to see if we could elucidate how unexpected this result was. So we have these plotted. This is just the chemotherapy arms. So we see PD-1 high in red is PD-1 high chemotherapy on Danube. Green is PD-1 low chemotherapy on Danube. So red is above green, but also blue is above purple. This is the PD-1 high versus PD-1 low chemotherapy in IMvigor 211. So this difference, I think, in the whole development program was assumed to not exist, right? That this biomarker target was predictive for the drug. It was not prognostic for the population. So we looked at the chemotherapies that were enrolled in these trials, and we looked at historical studies. We found roughly 11 historical trials that we could use, and we tried to see what was with that 10% null hypothesis, was that correct? And we know that these historical chemotherapy studies, they're mixtures of biomarker populations, right? They haven't actually, ascertained whether a patient's PD-1 high. This is all comers. If we look at, actually our meta-analysis says 10% right on the target of what we see historically for chemotherapy. But if we split this out by PD-1 high and PD-1 low, we see this big difference here. PD-1 low, it's right on that target. PD-1 high, it's actually statistically significantly above that target. And this is just chemotherapy group, right? And we're taking the 211, IMvigor 211 study here. We looked at survival. So we digitized the survival curves from these studies. If we didn't have survival curves, we used exponential. We came up with a kind of hierarchical distribution for survival. We looked at chemotherapy overall. It's right on top of that expectation. But we see that if we add on chemotherapy PD-1 high, we get this purple line, giving us a little more evidence to suggest that this is actually a prognostic marker for this population, right? It may not be predictive. And, I think that could be controversial, but that's kind of the conclusions that I come to. Now, this list is growing of drugs in precision oncology settings that have been removed from accelerated approval. There's roughly 16 in the last few years where we've seen this happen. So it's not just Atezo, it's not just Durvalumab. This is widespread. And I think what's happening is we still have this issue with distinguishing prognostic from predictive targets. So the question is, and we've seen modifications in regulatory policy now. There's kind of an overhaul of what they want to call accelerated approval, how you arise at that. They now want accelerated approval to be an interim analysis of a larger randomized study. And that interim analysis can be an evaluation of objective response rate and duration of response, or PFS, but you need to have that overall randomized trial for survival ongoing, is what the FDA is telling us. So we've seen Project Frontrunner come in, again, trying to push randomization earlier in the process, right, is how the FDA has responded to this. But I want you to think about, okay, so what happened here and how does real world evidence play a role? Because these are the issues we're actually facing in precision oncology. Could we have used real world evidence, right, to do something about this? Could they have understood that perhaps that target, patients may have outperformed the historical average if we would have been able to understand their biomarker status? And for that, so I want to go to the Friends of Cancer Research collaboration, their version 2, which actually looks at methodological recommendations for oncology case studies. I think I respect the people that are on this panel, and I think they make some really good points. I'm going to go back to my kind of considerations, and I want to ask the question, if we look at these oncology settings, do we have prognosis? Do we understand it? And if you look at Friends of Cancer Research, they talk about several fundamental issues with the real world data sets that they have. One, it's really hard for us to understand whether a patient is actually a first-line patient. That information is not necessarily captured, and they have come up with some criteria, no evidence of 180 days before diagnosis of a prior treatment, but that's pretty, maybe that's a good one. Obviously they thought of that, but it's not hard evidence, right? Histology is not universally collected in these EMRs based on the review of the Friends of Cancer Research group. So that's a problem, right? If we don't know if a patient has adenocarcinoma, for example, or whether maybe the histology of a liver metastasis, right? If we look at eligibility criteria, kidney function and performance status were often not included, or collected in these sets. It was difficult to ascertain this from real-world data sources. They see that prior drug regimens are often misclassified, and patients, there's a question of whether patients that had targeted therapies actually were measured for the targets in ALK and EGFR. So I would say we haven't resolved this based on what I see. What about endpoints? Well, survival is clearly the most objective endpoint that we can use overall, but also in the context of real world data. And they do talk about that, and that there could be some bias in survival, but that's kind of what you want to use. If you want to look at progression-free survival, they suggest we look at time to change in treatment, basically length of time from date of treatment initiation to the next systemic treatment. It's very important to understand that patients are not scanned at regular intervals in the community. My understanding is that patients are scanned only when necessary. Patients are not biopsied at with regularity in the community. Some are too unhealthy to be biopsied. They're biopsied only when necessary. That's fundamentally different from a clinical trial where we have a very specific schedule for evaluating, tumor burden and how it's changed. So we have to adopt this time to next treatment. We can also look at time to treatment discontinuation, which could be a combination of progression, but also adverse events, right? Adverse event risk. And they say maybe we can use that as a surrogate for PFS. They say that we don't have tumor progression. There isn't an analog for RECIST criteria in the real world databases. Now, you know that there's been monetization, people claiming they can use AI algorithms with annotations from clinicians and come up with a criteria for RECIST. So I don't know what your view is on that. I think that would be quite challenging, I'll just say. If the patients are not scanned at regular intervals, I don't think we can use those clinical annotations and come up with something as precise as RECIST. So I would take that off the table when we consider this. So I think there's big questions about the endpoints as well. What are other headwinds for oncology? Well, I don't know if anybody's ever tried to take clinical trial data in precision oncology context and match it to something like the TCGA. I have done that, and I can say it's very difficult. So I helped advise Bayer on an analysis that was done to extend larotrectinib actually to the Canadian approval. MD Anderson had 77 patients with NTRK fusions that they treated. The other sites provided very few patients because MD Anderson's kind of ahead in this context of identifying patients. But the idea was, we could just go to the Cancer Genome Atlas and we can grab patients from the indications we observed in this tumor agnostic study, put those together, and we can answer the question of whether NTRK fusions are a prognostic marker, right? Because the question was, these are single arm studies, and again, they wanted to understand, does having an NTRK fusion, put you at better prognosis or worse prognosis? So that was the idea. We can use these databases. We can use TCGA. I went to TCGA and, oh man, it is a mess. So actually, it just so happens at this MD Anderson study, the majority of patients are thyroid or salivary gland cancers, right? This is the breakdown of these 77 patients with NTRK fusions. The rest are, very sparse in these other indications. And it turns out there's very few thyroid or salivary cancer patients in the TCGA. Moreover, if you look at prognosis across these indications, it's vastly different. So, I would say that was an unsuccessful attempt to understand the prognostic nature of NTRK with real-world data. Other challenges is the data, so I've been a part of some major medical centers that have massive investments in clinical trial infrastructure, in data infrastructure, in research nursing. And they are able to accumulate data, but I also know the limitations of their data. What about the community clinic? In the US, we have community clinics that I've been told they can treat up to 40 patients a day. Are they really capturing, without all that infrastructure, are they really capturing reliable data in their EMRs? I would ask that question. I do know that things like T-stage, end-stage histology and line of therapy, even the big academic medical centers struggle to always capture these in their EHRs. Now, there's been investments to move that forward, but that still exists. What about patient identification? We do have FDA-approved targeted therapies for several actionable driver oncogenes, including EGFR, ALK, ROS1, BRAF, MET, RET, KRAS, G12C, and NTRK. This is just for non-small cell lung cancer. But in order to find these other targets, you have to go through NGS testing, next generation sequencing. So really the question is, how good are we doing at identifying these targets? Now we have FDA approved drugs. We just wrote a paper in JCO Precision Oncology that evaluated this. We found that, and this was a couple of years ago when we collected the data, about 60% of academic practices in the US are using these multi-gene panels. So they're able to detect these more rare targets, while only about 28% of community practice. Most patients are treated in the community. They're not adopting NGS testing yet at a wide scale. So therefore, we're not even finding patients that have FDA-approved targeted therapies. How can we expect to understand if these are truly different subtypes of the disease? How can we expect to understand their targets when we bring real-world data into a clinical trial? You know, that's the question I would ask. And then let's think about these novel targets. Of course, the novel targets are not there, and clinical trials in precision oncology is all about novel targets. That is the story, right? It's not about we have the information in the servers and we just need computer scientists to solve the problem. No, these trials are run to test novel hypotheses about novel targets. Of course, those are not in the EMR. If we don't even have, you know, targets for FDA-approved drugs in the EMR. So if you want to see, there's additional issues. Even when we run clinical trials in oncology, there's now a question that we don't really understand how to put these endpoints together: response rate, progression-free survival, overall survival. You can see articles about irreconcilable differences. And this is clinical trials before we even talk about the issues with real-world data. I do think there are some case studies where this could be successful, however, and I'm working with Dr. Flowers at MD Anderson, who runs this LEO consortium. They're doing a very careful job with accumulating data that is necessary that they could use to inform trials. There have been trials like this that have been run in lymphoma. You can see they've taken EHR cohorts, they've combined those with single-arm cohorts, and they have used a proposed methodology for integrating these. In the interest of time, I'm going to fast forward these. These are here for your reference, but I've highlighted kind of examples where they did this, so you can go in and see exactly what they did here. Of course, it gets down to, you know, causal inference at some point. You know, what have we acquired to believe that we have done a good job with measured confounding? How are we going to basically integrate that data together, acknowledging that there is confounding, there is bias, but we can attempt to adjust for it, right? That is the crux of all of these. In lymphoma, it's very hard to study overall survival. So that's another reason why they're doing this, especially follicular lymphoma, where patients live for 20 years. So, okay, but if you look at the FDA approval, the basis for approval based on this study, nowhere does it reference the real-world cohort. In fact, they're referencing just the single-arm results for the active cohort. So we do have the academics telling us that this was a foundational thing that we did with bringing real-world evidence in. But then we have the regulators not even referencing the real-world evidence, and I think that's very interesting. I think it's very telling. There's issues here with in the U.S. trying to have better representation of clinical trial populations moving forward. And I would submit to you that people suggest that real-world evidence is going to be a way to overcome that. But I don't think we've solved that problem yet, because all of the statistical methodology is about discounting patients that don't look like the trial patients. So if you're telling me that trial patients are not diverse, and then we control bias by, you know, basically reducing the influence of patients that don't look like the trial patients, I don't think we've solved this yet. And I think what we need is some sort of suitability score for these external cohorts that can tell us about what is the extent of unmeasured confounding. Why do we believe you captured the appropriate variables that we would need to adjust for? And I have some ideas on how to do that I've shared with the FDA and other partners, but I think this needs work. So if people want to work in this space, focus on this. Like, what do we do about the unmeasured confounding? FDA will not be convinced until you can tell them something about this, in my opinion. Okay, so let's go back to this well-controlled clinical investigation. I will leave you with, you know, this ODAC meeting that just took place in October. If you like these topics, I would encourage you to watch this 6-hour ODAC meeting where, you know, the FDA kind of scrutinized a company that had just run an open label randomized study in a precision oncology setting. The prior single arm study was very promising. It was a target that had not been, it was a subtype that had not been targetable before. So there was a breakthrough. Those patients with that genetic subtype had no treatment options. They had a very promising phase 1 study. They went to randomize with an open label study. And there were issues with patients dropping out of the chemotherapy arm after they realized they were in the chemotherapy arm, right? So they're excited to get this targeted therapy for their target. They get in this open label trial and they drop out when they find out that they've been randomized to chemotherapy. And just to show you the extent of this happened in roughly 13% of the chemotherapy arm and only 1% of the active therapy arm, right? And there was another issue with kind of understanding progression between these two arms. But let's put this into context. You're talking about 174 patients and you're talking about 23 that have dropped out, right? Now, the FDA ODAC meeting voted 2-10 that this was not a well-controlled trial because of this. And, you know, I think they're pushing against these open label randomized studies in oncology. There were some other issues with, they said, confounded the evaluation of response. But I would submit to you, based on what I heard in this ODAC meeting, that we don't have this continuum here. But this is a wide gap between the traditional randomized trial and this hybrid controlled trial now. Because if 13% of your controls can drop out with informed censoring, and that completely invalidates the result of a randomized study that doesn't incorporate historical controls, we can imagine how much less well-controlled these external controlled trials are. So to me, this kind of calls into question the premise a little bit. And okay, I'll stop there. Now I ask you to vote again after this presentation. I've kind of debated with myself, I guess, but eager to hear your thoughts and your questions.
So can we go to the poll, Kelsey? I think you can launch the poll if you could. Yes, thank you. We'll see if things have changed and in what direction.
Should I stop sharing screen or?
I think you can keep your screen, Brian. You will not see the results, but I will read them to you as I did before, so we can discuss any differences between the first vote and the second. And thank you for leaving a few minutes for questions. We have a few. I think we can go to the results. Hopefully people have had the time to vote. It's an easy question. And what do we see? Okay, Brian, you don't see this, but the results are now, yes, agree, real world evidence will play a transformational role in future registrational trials. Now, 39% of attendees have said yes, 34% have said no, and 27% have said, I don't know. So I mean, the good thing is that the number of don't know has gone down, and I will let you comment on the other results. 39% yes, 34% no.
Yes, I guess my objective was to point out kind of all of the issues that one would need to consider to effectively do this and ask the question, how do we resolve these issues? So I'm not kind of pointed in one way to the other. I would say that if you can fit my criteria that I gave you at the beginning, then I would say it's useful. I would submit to you that this criteria is easier to fit outside of oncology on the basis of what I've seen. And, you know, going back to these examples of withdrawn drugs, we would really have to have some natural history cohorts that are studying these novel targets in parallel with the clinical trial to integrate that evidence. So, yes.
That's a question I had actually, if I may. The question that I'm not sure that you can hear me. Yeah, the sort of claims that Dr. Boudet made, you know, that you no longer need randomized trials, you can just approve drugs based on real-world evidence. I was wondering, when a new drug comes, you know, to be tested in clinical research, like atezolizumab, you showed that example. obviously you have no data on that new drug in real world. I mean, the only way to get data in the real world is to get the drug approved. So I just cannot understand why he claims that you can approve drugs before you have any data on these drugs. So could you elaborate on that?
Yes, I would say that there's a lot of interest in improving the efficiencies and reducing the cost of drug development and healthcare overall in the United States. And I think the computer scientist community is framing the question in a way that they have the solution, right? And I think it's quite disingenuous because at the same time, the FDA is telling us that we don't do a good enough job with clinical trial enrollments, right? We need to have better representation of our population in clinical trials, not just people that are wealthy enough to fly to academic centers, right? So that is at odds with this idea that we no longer need these clinical trials, right? So I think it's harmful to go out and say this, to the public in this way, because the reality is we need more people to be engaged in clinical trials, and we need more people from diverse backgrounds to be engaged. So we don't want less engagement if we want to move the science forward. And yeah, I think there's a fundamental misunderstanding in that community of what clinical trials are. And they are, and I would add on to that as well. If you've ever worked with academic translational labs, the culture is so different, right? Those labs are funded by a sequence of grants that absolutely have to renew to continue the grant. So I've never worked with a translational researcher that absolutely did not have the solution, right, to whatever problem we were working with. It's only when industry invests in a trial that we actually get to check and filter that enthusiasm. And you're not going to hear these guys that have research dependent on a particular mechanism working. That I question the objectivity of that research, and I feel that industry plays a fundamental role in filtering, right? Filtering these ideas. So that's also, I think, largely misunderstood by the public. So.
I got an interesting question about what you just said, that the cost of clinical trials has become almost unaffordable, even for big pharma companies. So someone asked the question: isn't the solution to combine the benefits of real-world evidence and randomized trials by making randomized trials far more pragmatic and just randomize patients as if they were going to a routine visit instead of having them enter a very complex protocol?
And I guess I would respond to you: listen to the 6-hour ODAC meeting and see how the FDA reacted, because you could argue that this trial had pragmatic elements. It was an open label trial. They did it during the COVID pandemic. They did the open labeling so that patients could take oral drugs and not have to come in for IV infusions. This had pragmatic elements, and it was absolutely scrutinized to a level I haven't seen recently. So these individuals at the Oncology Center for Excellence haven't pivoted to show a willingness to accept those pragmatic elements, even though they make slides like this talking about them. I haven't seen well, I don't know, Mark. What do you think, Mark? That's how I.
No, it's not my talk. I mean, it's your talk, so I don't want to give my opinion. But on the whole, I agree with you absolutely. Let me ask another question here, because the clock is ticking. A question about rare diseases, I guess. Someone asked the question: you mentioned the infeasibility of randomization, and the question is it ever infeasible to randomize?
That's a great question. So, I mean, one could argue that the excitement that happened with this drug, and I won't talk about it too much, but the excitement that happened when this drug was approved for a target that they've been trying to target for 30 years, somewhat made it very difficult to randomize. Because now, if you're aware that you have that target and you're going to this trial, you want this drug, right? Based on what you've heard, and you have a terminal disease. So it's not infeasible. It's not like you can't do it. It has been compromised in this setting. I would say that as populations get rarer and rarer, then the Cystic Fibrosis Foundation you know, this trial's moving forward in cystic fibrosis. We're talking about 5-to-1 randomization now. They want to hold on to a little bit of randomization. It becomes more and more infeasible as you have, you know, as you have a smaller and smaller population that you're willing to give a placebo, right? So I guess it's never infeasible, right? Maybe that's not the right word. It could be infeasible to blind. And so blinding and randomization go hand in hand. So blinding becomes infeasible in some settings, like the first setting, which I would say then impacts the credibility of the randomization. And then I would say, as the population gets rarer and rarer, you may just not be able to enroll populations into clinical trials because there's a small subpopulation anyway. The clinicians participating in the trial may be unwilling to randomize their patients. And I was on the data safety monitoring board at MD Anderson for 4 years. I saw many trials die because of enrollment.
Lack of interest. So, yeah.
Exactly.
Well, I see that we're on the top of the hour, so I think we have to cut it short, unfortunately, because we could have gone on for much longer. But again, thank you so much for this very interesting talk. And I will pass the word back to Kelsey to close the meeting. Thanks a lot.
Thank you. Thank you both. So we've reached the end of our Q&A and the end of the webinar. So please note, if we couldn't get to your questions, we'll be sure to follow up with you after the presentation. And if you do have any further questions, you can direct those to info@iddi.com. That's info@iddi.com. And you'll also shortly be receiving a follow-up email from us with access to the recorded archive for the event, as well as a survey window that will pop up on your screen. And we appreciate your participation, as always, to help us improve on future webinars. So thank you, everyone, for attending. Thank you, Dr. Brian Hobbs, for your very insightful and engaging presentation. And thank you, everyone in our audience as well.
Thank you.
All right. Have a great day,
Thanks
everyone.